Today we have two fellows speaking, each presenting about half-hour-long presentation. Our first fellow is Christopher Katalinas. He's a member of the 2017 Executive Knauss Fellow class and works for the National Sea Grant College program. Chris grew up in Manchester, New Hampshire, and went to Dickinson College for his Bachelor's of Science in Biology. After that, he went to the College of Charleston to get his MS in Marine Biology. And a fun fact about Chris is that he ran for the track team in college and competed in the decathlon, specializing in the pole vault. However, he highly advises not using what appears to be sturdy bamboo as a pole vault on the beach. Love to hear more about that story, but that's for another time. So let's welcome Chris.
Thanks, Megan. So this is going to be kind of a combo presentation. I'm going to focus on my master's thesis research, but kind of tie in some things that I've learned in terms of communicating science through my fellowship this year. So my master's thesis was focused on informing red drum stocking strategies in South Carolina. So red drum, if you aren't aware, is a popular recreational species all along the Atlantic coast and in the Gulf of Mexico as well. And so in the early 90s, populations of red drum in South Carolina had declined due to overfishing. And so the Department of Natural Resources began a stock enhancement program to serve as an alternative to restrictive management to try to increase the population of red drum so more recreational fishermen had more fish that they could capture. And so it's important to note that recreational fishermen can take juvenile red drum within a slot limit, but adult red drum, they had to release them back into the water. And so it really focuses on the juvenile population. So red drum exists all on the Atlantic coast in two genetically distinct stocks. And so my study focused specifically on the southern stock in the Charleston Harbor estuary. So juveniles live within the estuary, and then once they reach sexual maturity, they move offshore. And so juveniles are spatially separate from the adult population. And so when you release fish, millions of fish in some years, that are all pretty closely related, you have the potential for reducing the genetic diversity of the wild population. And so my study was specifically looking at, is this happening in the population associated with the Charleston Harbor estuary? And so ultimately what I found is that stocking managers can have as few as 10 adults in their hatchery. And then when they go and sample the wild population, 30% of those fish could be from the hatchery, and there will be no changes in the genetic diversity of the wild population. So this ultimately achieves this balance of releasing fish into the wild to try to increase those numbers that are available to recreational fishermen while minimizing any potential changes in the genetic diversity. And I looked at genetic diversity because it is a measure indicating the adaptive potential of a population. So if you have lower genetic diversity, over time the population is more at risk to a variety of factors, whereas if you're more diverse, then you're more resilient over time. So why did I have to look at this balance? Why did I have to look at the genetic impacts of stocking to begin with? Well, in the hatchery, stocking managers are limited by resources and space. And so I'm not a very tall individual, but my head was at the top, so the water line is just a little bit below my head. And so you can really only fit 5 to 6 adult red drum in a single tank. And they have a stocking program for multiple species in South Carolina, so they can only devote 2 to 3 tanks to this fish species. So it was important to identify, well, what is the minimum number of adults that you can have in the hatchery in order to minimize your risk of reducing the genetic diversity of the wild population. So in order to identify this minimum of 10 adults in the hatchery, maximum of 30% of the population can be stocked fish, I developed a model. And so I incorporated known life history and genetic information into the model. And so I'll kind of walk you through what happens to fish as they move through this model. So they start out as age 1. Then they get a platter survival rate, so a portion of those fish survive to the next age class. And they move through the system until they reach sexual maturity and move offshore as adults. These adults then reproduce and keep that chain going. So this happened in the model for 90 years to allow the population to stabilize. Then I took adults from the wild population, brought them into the hatchery. They reproduce, and those stocked fish were then released into the same system that the wild fish are in. So they moved through their life stages, and then the adult fish joined the wild fish offshore. So those can spawn together. And then ultimately that population then feeds into one now kind of hybrid population where it's wild fish and offspring of stocked fish that have made it into the wild population. And so this continued over and over for, I ran it for 90 years. So something to note about red drum, they live to be about 45 is what their maximum age is estimated in the southern region. So they're a long-lived species and they exhibit overlapping generations. And so I essentially ran the model for 2 lifetimes for red drum. So this is all well and good. I ran this model, but what sort of confidence do I have in this? Ultimately, when you run a model, you want to be able to compare it to something that you observe in the wild, because otherwise you're just getting a bunch of data and it's you kind of look at it and say, okay, well, what does this mean? And so the first part of my study measured the genetic diversity of the population of red drum in the Charleston Harbor estuary. So the same area where we had stocked fish into for over a decade in South Carolina. And so to orient you to this, so on the y-axis we have 2 different measures of genetic diversity. I focus on total number of alleles and expected heterozygosity. These are different measures of genetic diversity, but they're some of the more common ways of measuring it. On the x-axis, we have the year. So this is the burn-in or stabilizing period. So this is 90 years where I allowed the model to run to allow the population to stabilize. From there, I started stocking fish from 1999 to 2011 to mimic the actual years that fish were stocked into the system. So these boxes with tails on them represent the results from the model. And then the blue squares represent the measures that I had observed from actual fin clips that were obtained in the genetics lab. And so these are measures of genetic diversity that we observed from fish in the wild. And so you can see there's pretty good agreement between both simulated and observed measures for both total number of alleles and expected heterozygosity. So this gave me confidence in saying, okay, well, when I look into the future, those measures are likely indicative of what we would expect under these different conditions for the model. So I want to take a step back for a second and kind of talk about some of the things that I've learned through the fellowship in terms of communicating new science. So you might have seen that the way that I've framed this talk has been a little different. I kind of gave you the conclusion pretty much right off the bat. And so to kind of go through this, so when you're communicating science to just a broad audience, an audience that doesn't necessarily have the same background as you, it's good to start with the so what. Well, why do I care? So, for example, red drum populations were declining, but it's a popular sport fish. And so we care about having a healthy, abundant red drum population for the fishing community. The takeaway message here is that stocking managers can make a 30% contribution of stocked fish. So when you sample the population, 30% of those can have come from the hatchery, and they can have as few as 10 adults in the hatchery in order to minimize the risk of a change in genetic diversity. And then from there, talk about the supporting details. Well, most important, know your audience. So I realize I'm standing in a room with a bunch of scientists, and you say, okay, great, Chris, 30% contribution, 10 adults. Well, what support do you have for that? So here are the results from my simulation. So it's the same x-axis, although this is generation time now instead of per year. So we had that stabilizing burn-in period. I ran it for 5 generations of stocking. So these horizontal gray bars indicate the stocking period. First one indicating when stocking began, second one when stocking ended. And then I ran it for another 5 generations to see any lagging effects. So after you stop stocking, what happens to the population? So these 3 rows represent 3 different measures of genetic diversity. We see the total number of alleles, again, the expected heterozygosity. Then I also included the effective population size. That's another commonly used metric for measuring genetic diversity. And so then in this first column, there were only 2 adults that I had in the hatchery. What happened to the genetic diversity over time? 6 adults in the hatchery in a 1-to-1 ratio, so 3 males, 3 females, and then 10 in the hatchery. And then each of these lines represents a percent contribution of stocked fish. So the top line, the solid one, is 30% contribution. The dashed line is 60% contribution. And the dotted line is a 90% contribution. So if you went out and sampled the population, 90% of those fish came from the hatchery, what would you expect to happen to the genetic diversity of the wild population over time? And so we can narrow in on the far right column with 10 broodstock. With total number of alleles and expected heterozygosity, one of the things I found was red drum tends to be really resilient to changes in this, primarily because of their overlapping generations and their long lifespan. And so actually for both of these, all 3 contribution levels never exceeded a threshold that I set. So there have been some past studies that look at if you maintain at least 90% of the initial genetic diversity of the population, so after this burn-in period, if the population never maintains 90% of its original genetic diversity, then that will likely indicate that it maintains that adaptive potential over time. So it will be more resilient to a variety of factors. And so one of the things that was really limiting when it came to determining how many stocked fish can be in the wild and how many broodstock do you need was the effective population size. And so similar to the 90% genetic diversity, there have also been studies that say as long as you maintain above 1,000 individuals for an effective population size, then that's maintaining the adaptive potential. There's been some debate around that, but that's where I held my threshold, was 1,000 individuals. And so that 30% contribution never drops below 1,000. And so that's how I ultimately came upon this balance of you want to increase the number of fish that are out there for recreational fishermen, but you don't want to negatively impact the population because ultimately you want to maintain a healthy population so people can continue to fish for the long term. And with that, I'd like to thank you all for listening, and I welcome any questions.
So knowing like the variability in survival, particularly of juvenile fish and estuaries, can you explain your thought process or what you did for building the model, the mortality that you built in for the, would you just use the same one as natural versus stocked?
Right, so the question was about how did I determine the survival rates and whether they differed between the natural versus stocked fish. So they were the same. There were past studies that showed no difference in survival between the two. So prior to this study, they had done a lot of research that looked at, are stocked fish actually surviving in the wild? Is it stock enhancement even feasible? And then looking at survival differences, because if they're raised in a hatchery, they might show domestication effects. They weren't in the hatchery for that long. They were in the hatchery for a maximum of 60 days before they were released into the wild after hatching out of the eggs. So I assumed equal survival, and I based it off of the CDAR estimates for the southern portion of the stock.
So your results in terms of minimum number of fish and then the percent of the stock population, How close is that to the way it's currently been?
Well above. So the question was, how close are my recommendations in terms of the balance to what is actually occurring? And so the greatest contribution that they've seen for the adult population is a lot higher in the estuary, but the proportion of adults that are stocked fish that actually make it out there is lower. But the highest year was around 12%. Of the population, and so 30% is still giving a lot of wiggle room there. And most years, in terms of the number of adults that actually contribute in the hatchery, because it's difficult to get all of the fish to contribute, at least in the latter portion of the stocking program, was around 10 as well. So that's also why I based those numbers off. Libby.
Sure. So is there any attempt to maximize heterozygosity in the hatchery?
Yes, there is. So the question was, are there any attempts to maximize heterozygosity within the hatchery itself? So they only keep adults in the hatchery for a maximum of 3 years, and then they rotate fish around the spawning tanks as well. So after they exhaust spawning pair, then they will release that fish from the hatchery and they'll bring in new fish. So they attempt to.
Are those fish being collected from different parts of the range? Or?
They are all being collected offshore of the Charleston Harbor estuary. And so there's the way the lab works is it splits up into the stocking program. There's the inshore monitoring program that collects adults as well. And it looks at the contribution of stocked fish to the natural population. And then there's a population genetics lab that processes all of the genetic fin clip samples. And so all of the adults that are used in the hatchery are from the local wild population offshore the Charleston Harbor estuary.
So this is sort of tangentially related, but curious about the economics of the hatchery program. So, like, who runs that, who funds that, and just more about the details on that.
Sure. So the question was asking for more details on the hatchery program itself in terms of who funds it, who runs it. And so the Department of Natural Resources is the one that runs the hatchery. And I believe that most of their funding comes from the state, but it is a compilation of researchers that pulls in outside funding sources as well, mostly from the genetics side of things. So they kind of work in a collection of 3 labs. And so depending on what research project they're doing, because it's a research scale hatchery, and so depending on what they're focusing on, whether it's seeing if those stocked fish are actually surviving, whether it's trying to increase the genetic diversity of the broodstock within the hatchery itself, it kind of depends on where the source comes from. Most of it is through the Department of Natural Resources. It has been operational since they started stocking red drum in 1999. So. Mm-hmm.
Your plot for the genetic diversity you saw from the genetic samples dropped in 1 year drastically.
Yes.
Do you know why that was?
Sample size. That was 7 fish.
Okay.
So the first couple years, so for those on the webinar, the question was, what was happening in this 2007 year that caused the genetic diversity to drop? And so it's driven by sample size. And so from 2007 to 2011, the sample size was 7 to like 20 something, whereas they were closer to 100 in the first 3 years. So it was cool to see that the model results mimicked that, because when I calculated these, I based it off the same sample size as well in the model versus the observed. So it was cool to see that even with a sample size of 7, I was still getting that match. Great.
With hatcheries and the life history of drum, so are there other people augmenting drum populations throughout their range, or is this just a research program? Or how many more hatcheries would it take? actually increase the population?
So the question was asking if there are other hatcheries that are trying to utilize stocking for red drum throughout their range. So the only other region that I am aware of that is doing this as heavily, probably more so heavily, is in the Gulf. So I know Texas and maybe Louisiana, primarily Texas, has a similar program that's on a larger scale. I want to say somewhere in the Mid-Atlantic, but not to the same sort of extent, and not for the same length.
Any questions? All right, let's thank Chris.